So question 1 asks, as described in box 5.1, I think it's pronounced Lewy, something like this. Anybody know better than that? It's German, I think. He was perhaps the first to demonstrate that synapses often communicate via chemical, really that should say chemical signals. Sorry. Let's walk through this experiment using scientific methods. So the hypothesis is that synapses communicate via chemical signals. I gave you that. And then he isolated frogs from he isolated frogs, and then he isolated their hearts as well as their vagus nerves. And he got 2 frogs, A and B. So who would like to give this one a shot? Go for it. Yeah, so he stimulated the vagus nerve, right? So somehow or another, he's stimulating it. That's supposed to be action potentials going down the vagus nerve. And that caused the frog A's heart to slow down. And that's, if you know a little bit about autonomic nervous system, which you don't really have to for the class, but vagus nerve is the parasympathetic output down to the heart. So if you stimulate that, that's going to cause the resting and digesting parasympathetic nervous system, which is sort of like the contrast to the sympathetic nervous system, to stimulate the heart, which then in turn causes heart rate to slow down. So what did he do next? Yeah, so he got a little syringe and he sucks up the saline solution that was surrounding frog A's heart, and he sent it over to frog B's heart, which is kind of interesting. So let's keep going with this. And so what did you put for the control trial? Like what did you put for that? Yeah, so he'd need to do something different here. Let's come back to that one, because so the prediction I think is sort of the key thing. So if this hypothesis that these synapses are communicating via chemical signals, and you stimulate the vagus nerve, you pull out solution then from frog A, what should happen when you inject that solution on frog B? Yeah, because it's got, ideally, hopefully, you've got enough of those chemical signals in the saline solution you suck out of frog A's pericardial cavity, so that when you inject it around frog B, or into frog B's pericardial cavity, those chemical signals will then be transferred to frog B, and its heart should slow down. Does that make sense? So you're capturing chemical signals and moving it from one frog to the next, and that should do it. And what happened? Is that, in fact, what happened? As far as results, sorry. Yeah, it slowed down. So it was a successful experiment, which would mean, I'm just going to sort of finish it, that means that the hypothesis was supported. So our prediction matches our results. We expected the heart rate to slow down. It did slow down, so that's supporting this idea that synapses communicate via chemical signals. Did anybody put anything different or additional? I think the control thing is kind of interesting in that, this is one of these experiments. The fact that it worked makes it pretty nice evidence that these things are communicating by chemical signals. Ahead of time, I would not have anticipated that this actually would work. To me, this would be such a shot in the dark where you'd expect to get enough of those chemical signals. So what's happening here is vagus nerve is releasing its neurotransmitter, which is actually acetylcholine, onto the heart. So you have to capture enough acetylcholine and then move it over to frog B and then inject that onto frog B and have enough of those acetylcholine molecules around hitting the right spots on the heart that it could actually cause the heart rate there to slow down. So that's kind of an amazing thing, I think, that it even worked. So if it hadn't worked, if the results had not been that the heart rate slowed down, to me, this wouldn't be all that much of a problem for the chemical hypothesis, simply because it seems like there's a large probability that you just wouldn't get enough of the acetylcholine. But it worked. It actually did work. I think one thing I would add as far as controls is that I think you would want to make sure that the reason that frog B's heart slowed down was due to the acetylcholine that you captured per se, and not the fact that you injected saline all over it. You know, that would be sort of an alternative explanation. So I think one thing you certainly would also want to do is you want to have a control where you're just injecting blank saline, the same saline that's over here, except without the acetylcholine that you captured. So you could even pull it out of a frog's heart before you stimulated the vagus nerve to release all this acetylcholine and inject that saline, and you should expect no effect then on the heart rate. You got to make sure that the saline isn't what's causing the effect. It's got to be the acetylcholine that did it.
Was this result like easily reproduced?
Yeah, it did work. I mean, they did reproduce it, and it was published, and it was the first, I should say, good evidence that some synapses at least communicate by chemical signals. It was not neuron to neuron synapse in this case. This was neuron to heart, but still, it was a replicatable experiment. The story is great in the book. Like, it even talks about how if he'd been actually awake, like the story is he woke up in the middle of the night to do, like, thinking of this experiment, ran into the lab and did it in the middle of the night. And the argument is that if it had happened during the day, he probably wouldn't have even tried it because it seemed so freaking absurd that it would work. But it did. So sometimes it's worth it to try absurd things. Is that okay? Any questions? We should keep going. All right. Explain the difference between spatial and temporal summation, making explicit reference to figure 519. So let me get to figure 519. And I'm thinking, so we're on an odd chapter, even odd. So how should that work? So we should go clockwise? I don't think we've been going this way. So let's go this way. Because you started, so like, can we have like the next row? There's a group there, right? Let me pull the figure though. You can get your thoughts in order. 519. Okay, so go ahead and, I don't know who wants to talk. Hopefully somebody who hasn't yet, but if everybody has, then you can just pick. I don't care. Awesome, thank you.
You said spatial summation is when two or more presynaptic inputs are active at the same time, and then their individual potentials are added together. And then temporal summation is when the same presynaptic fiber fires multiple action potentials quickly.
Okay. Great. So okay. What did you put about the quantal analysis stuff? That's the.
So that it's. Well, you read that in the central nervous system, like only a single synaptic vesicle is released with an action potential, or like some small number. So they need the summation, so more vesicles are released that release more neurotransmitters. So there's a stronger action potential.
Right. How does that contrast with the peripheral nervous system?
So like the peripheral nervous system, it said that multiple or like almost 200 synaptic vesicles are released, so there's enough neurotransmitter to get the action potential.
Any additional comments or anything about that one? So, I mean, the figures are pretty nice. So again, the idea is with spatial summation, you're summing across space. So that means you've got multiple dendrites coming in, or sorry, multiple axons coming in on the same presynaptic cell, and they could be hitting the same dendrite or multiple dendrites, but they're all arriving simultaneously at the. And so you're summing across the space of all the dendrites. So it's called spatial summation. So you're summing across space. And so in that case, all of these basically add up on top of each other. And they're kind of piggybacking on top of each other such that you get a very large excitatory postsynaptic potential now because they're all basically summing together. And that's in contrast to just getting one, which is what's shown in this figure in part A, where here's one action potential from one presynaptic axon. It's a relatively low EPSP, but if you get multiple, they all sum together. And you can't really see them, but you can sort of imagine they're just stacked on top of each other, basically. Whereas in temporal summation, you only have one input, like one axon delivering signal, but you get a barrage of action potential after action potential after action potential. And with each action potential that arrives, you get a new wave of vesicles with neurotransmitter being released into the synapse, and therefore a new wave of excitation down here. So you kind of have one little blip, but then even before that excitatory postsynaptic potential would wear away, you have another one that basically then sums on top of it, and another one on top of it. So here you're summing not across space, you're summing across time. And so we call it temporal summation. And the key thing about the central nervous system synapses that was discussed in your book, this idea of quantal analysis, is where you're trying to figure out basically how much of a EPSP, or could be an IPSP, an inhibitory postsynaptic potential, but how much effect does one individual vesicle have on the postsynaptic cell? Because really it's those vesicles, like each vesicle is kind of a unit of information that's sent from the presynaptic cell to the postsynaptic cell. And the whole idea of quantal analysis is to try to understand how many vesicles are being released, how much influence does each individual vesicle have. In the central nervous system, you often are only releasing one or two or a very small number of vesicles from the presynaptic cell in response to each action potential that arrives at the axon terminal, which means that each vesicle is only a small amount of neurotransmitter. Each batch of neurotransmitter can only really cause a very small EPSP. It could literally be like 1/10 of a millivolt. So in order to get up to threshold, in order to go from -65 up to -60 or so, where you're getting up into the threshold range and where you could start to open sodium channels, you have to have summation. Otherwise, you just won't fire. And that's really, if you think about it, that's what brain neurons, central nervous system neurons do, is they're taking inputs from all over and combining them. And it's only when you get sort of a synthesis of information from many different inputs, or a very large signal from a smaller number of inputs, that you're going to get up to threshold and fire the next cell down the line. So that's really what we're seeing here, is we have to have these sort of summative effects. But it doesn't matter at the neuromuscular junction, because in the neuromuscular junction, there's just a crazy number of vesicles released. Like you said, it's like 200, which always is going to get that postsynaptic cell. In that case, it's the skeletal muscle, but it always will get that postsynaptic cell up to threshold every time there's an action potential that gets down to the end of the motor neuron. So it's very reliable, but it's a different kind of function. There, you want to start causing the skeletal muscle to do something every time a signal gets down there. You're not trying to integrate information, you're just trying to respond. And so those neurons are set up in a different way because of it. Any questions on that? Is that okay? Not too bad. You don't have to understand. Like, it talks about in the book how they do this, where they try to look for these little mini EPSPs that just happen randomly because you get random spontaneous release of vesicles. I didn't really get into that, but I do want you to understand that the idea about quantal analysis is trying to investigate how many vesicles are released and how much one vesicle does, and how that plays into our understanding of summation. Okay, here's the good one. So number 3, so we did that one. Can we do you guys? Is that okay? I don't know who wants to go, but you'll go? Okay, great.
So for A, we said that just the description of line constant is the point from the depolarization to the origin. And this is important for efficiency of transmission down the dendrite, and also the rate of transmission.
Yeah. I can never spell propagation. I always put an O every single time. All right, important on the dendrite. Oops. So it's velocity. So what'd you say on this one? So how will increasing the internal resistance affect the length constant?
So that it would slow the length constant.
Resistance should slow, so reduce, you mean? Okay. What about membrane resistance?
It should increase, if the increased memory resistance, or with increased memory resistance, the length constant will also increase.
Okay. And then this last one, what'd you say put for this guy?
We said that, like, increasing conduction, is like related to the increased internal resistance and membrane resistance, relating back to like the efficiency of the conduction speed.
Let me write this down. Do you remember any specific examples that we talked about in chapter 4 that were?
Giant squid.
Okay. Actually, I'm going to. It's actually normal squid, but they have giant axons.
Yeah,
But
yeah.
it's all right. Yeah, so squid giant axons. So what do squid giant axons do? So they're really, really large, right? So what does that mean for these things we're talking about?
There's less resistance because there's more room.
Yeah, it's less internal resistance, right? So we said increased internal resistance should cause reduced length constant. So if it's less internal resistance, they should have a larger length constant. Let's pull up the figure here. We need to talk about what length constants are with a picture. Shoot. That's it, right? Yeah. So the idea with length constants, like you said, it's this distance from this point over here to the origin. The origin is where you stimulated the dendrite. So the origin is where you have this connection, the synapse between the presynaptic cell and the postsynaptic cell that opened a bunch of channels and caused, in this case, an excitatory postsynaptic potential that got you all the way up to here. So this is a pretty decent depolarization. So that over here, that peak at the origin, that is 100% of the depolarization at the origin. Let me come over here so I can get the recording in. So that's 100% right there. And then what you do to measure the length constant is you basically just watch the signal decay. Because if you think about like transmission down a dendrite, there's at least no voltage-gated sodium channels to speak of, not enough to really matter anyway, that would be able to regenerate this signal to allow you to send the signal down a dendrite. So instead, what you're doing in this figure is you're basically you've found a spot where there's a presynaptic cell delivering neurotransmitter at a synapse on a dendrite. So this is supposed to be a dendrite that's then connected to a cell body, which then has an axon hillock and all that jazz. So the origin is here. And then what happens is that as you measure further and further down the dendrite, you see that some of the signal, some of the sodium ions that came in up here from the ligand-gated channels that you opened when you hit the neurotransmitter, some of those sodium ions will start diffusing down the axon. But as they do so, some of them are going to leak back out, whereas others are going to keep going. But because of this steady leakage that you're getting, plus the fact that some of them went the wrong way, and therefore they won't keep on going down this way towards the cell body, because of that leakage, you'll gradually have that signal decay and decrement over the length of the dendrite, such that by the time you get over here, you could be at a relatively low amount of signal that actually passed down the dendrite. The length constant is basically just trying to measure how quickly the signal decays. Okay, so what you do is you keep measuring down the dendrite, and you find a spot, and maybe it might be right here, at which the signal is now 33% of the original strength. So you're basically measuring how long do you get to travel down that dendrite before your signal is only a third of what it used to be. Okay? And that measurement, that distance, is indicating how efficiently your signal is traveling down. If your signal is moving quickly and you're not losing lots and lots of ions, you may be able to go quite a long ways down the dendrite before you lose signal, and therefore you might have a really large length constant. Whereas in contrast, if you have a really leaky dendrite or a really narrow dendrite with lots of internal resistance and the signal can't get down very quickly, then fairly, you're going to see over distance, you'll see that the signal decays pretty quickly. And in those cases, you'll have a really low length constant. So basically, the story is, as it said in parts B and C, if you increase membrane resistance, that basically is trapping your signal in the dendrite, which is where you want it. You won't lose signal over distance. So if you increase membrane resistance, this should increase your length constant. And anytime you increase your length constant, that's a good thing for your signal. That means you're getting more signal, so better signal propagation, or I forget exactly how I phrased in the question, but. Similarly, if you, really in contrast, if you increase the internal resistance, that's basically corresponding, the internal resistance is going to correspond to the diameter of your dendrite in this case. So if you have small diameter dendrites that have lots of internal resistance, that's going to cause a decrease in your length constant, which then is going to correspond to less signal transmission. Okay, does that much make sense? Any questions on that much? I used to lecture on these topics before you even had a chance to look at it, and then people would just like stare at me with like deer in headlights, and that still is happening, but I think you've had a chance to hopefully see it a little bit before we start talking about it. But this is the idea, is that you're just trying to quantify how long before you lose a third of your signal. And the further you can go without losing 2/3 of your signal, the more efficient transmission you're getting. And that's basically a function of your membrane resistance as well as your internal resistance. And we talked about each of these things in the axon chapter, because the same thing applies to axons. So here we're specifically talking about dendrites, and it's really important for dendrites because they don't regenerate their signals. You've got to get that signal from the dendrite all the way down to the cell body, down into the axon hillock down here, or else you won't fire an action potential, period. You got to, there has to be, that's an important thing to be aware of when you're talking about how dendrites communicate their signals. But axons have the same issues. There, though, it's not so much about whether the signal will get there. In the case of the axon, it's really about how quickly does the signal get to the next step down the line. And so we talked about internal resistance having to do with axon diameter. So if you have really narrow axons, like some of the pain axons have really narrow diameter, they have a lot of internal resistance, and therefore they send signals really slowly. So that means they have a low length constant and therefore decreased signal transmission efficiency. Similarly, if you myelinate, this won't apply to dendrites, but it does apply to axons. If you myelinate your axons, that increases membrane resistance because you're insulating them. That's going to result in longer length constants within the axon, which will increase signal transmission speeds because you're not losing those ions. They're not leaking back across the membrane. So it all is telling you the same story that we talked about in axons, but we're talking about dendrites mostly in this chapter, but it's all the same stuff. It's all membranes that are, and we're just trying to describe how well signals get down them. Any questions? I think that's the hardest thing on here. All right, well, let's keep going. So then we get into question 4. Question 4 is just kind of, we've been dealing with all kinds of channels, so let's just try to get them all in one place and track them all. So where were we? We were there. So I don't know. That'd be great. Yes, thank you.
So we said that potassium and sodium lead channels are both located all over, so the dendrite, the cell body, and the axon.
Okay.
And then potassium lead channels they're always open to allow potassium ions to flow through and help set the negative membrane potential.
Okay.
And then sodium lead channels do the same thing except with sodium.
So they're kind of making some positiveness to the membrane potential. But there's far fewer of them, so the potassium ion channels basically end up winning the competition. But the sodium does have some effect. You're not at potassium's equilibrium potential. You're somewhere in between, but more towards potassium's equilibrium potential. Okay. Voltage-gated sodium channels.
We said they're located in the axon, and they're used for action potential initiation and propagation.
Okay.
And then voltage-gated potassium channels are in the axon, and they help repolarize the membrane after depolarization.
Okay.
And voltage-gated calcium channels are in the axon terminal, and they help excite the neuron and release neurotransmitters.
Okay.
Ligand-gated cation channels, we said were in the dendrites in the cell body. They allow cations to pass through the membrane in response to a neurotransmitter.
Yeah, and then the chloride ones.
Yeah.
What did you put for that?
We said that they were located in the dendrite in the cell body, and they kind of do the same thing as the cation channels, except with chlorides, it's like all negative.
Yep. So chloride to enter. They're pretty much always inhibitory because that inhibition is going to, or the chloride ions cause the membrane to become more negative on the inside, and therefore they would cause inhibition when they're opened. They are sometimes found in dendrite cell bodies. Sometimes you have those axon-axonic synapses where one axon is coming in talking to another one, and in those cases they can also be found on axons, but they're at synapses that would be on axons. And that's the key thing, is both of those ligand-gated channels would be found on synapses. We didn't really talk a lot about chlorided channels except on the chapter 4 homework, I think, where we said what happens if you're at the memory potential for, your memory potential is equal to the equilibrium potential of a chloride ion, and then you start opening chloride channels. I mean, this would be that case, basically. And in that situation, if you're already at the equilibrium potential, you don't cause a change. But in all cases, you open a channel for an ion, it'll cause the membrane to shift towards the equilibrium potential of that ion. Let's see. We are almost out of time, but I think we can do a quick job on number 6, 7 here. So is there another group like right here? Like you guys are, right? So would one of you like to? Here's the figure.
Okay, so basically we said that we would inject nicotine into that area to block the nicotinic receptor, and then we would inject the atropine or the other one to see if the other receptor on the other organ, the target organ, was the same. If it was the same as the nicotinic receptor, it wouldn't react because the nicotine would have blocked it, but if it was a different type of receptor, it would react because it wasn't blocked.
Okay.
Does that make sense?
Mostly. The only thing is the nicotine will actually stimulate this one. So if you wanted to, so here's the thing. This gives you a toolbox. You've got these two different kinds of receptors, and I'm going to post little videos actually talking this out in more detail. But the idea is that you can deliver nicotine onto these receptors, and that will only cause nicotinic receptors to open, but these muscarinic ones won't. Muscarinic receptors would only open when you deliver muscarine. But in real life, and muscarine's a poison that comes from some specific kind of mushroom. The cool thing about this is that these are both acetylcholine receptors in life, right? In our bodies, both of these are responding to acetylcholine, but we have these other compounds, nicotine and muscarine, that will specifically only bind to one or the other of them. and they will mimic acetylcholine's effects in those cases. We also have these poisons that block them. So there's curare and there's atropine. I think curare is from poison dart frogs or something cool like that. Curare blocks nicotinic receptors, though. So if you wanted to know if something is a nicotinic or muscarinic receptor, you could administer curare, which should block any nicotinic receptors there, and then you could deliver muscarine or acetylcholine, and that should cause any muscarinic receptors to get stimulated, but you won't cause any nicotinic receptors to get stimulated. So you can sort of tease these things out by using this sort of combination of poisons. All right, so I will post a short video talking about that question again, and then there's, I think, 2 more, but I think the last 2 aren't that bad. So I will post that on Blackboard so that you'll be able to see that and refer to that, but at least you got to talk about it in groups.